Morning, everybody. It's been a whirlwind day and a half, but a pretty wonderful and awesome day and a half. So what I'm going to do in the next 5-10 minutes is really summarize where we're at and what does the future look like. Maybe. There we go. All right, so I think one of the things that we've achieved over the last couple of days is I think that we have a better understanding about what family priorities and needs are. And this is hugely beneficial for us as researchers as we go back to the lab and think about our research and how to design experiments. So in particular, one striking thing is that I mentioned when we opened yesterday that Stephanie did a poll for the Facebook group in terms of what are the biggest research priorities for you. And largely seizures and regression were the top kind of two priorities. And then Christina yesterday did that very cool tool that I told her I'm going to steal because I think it's a great way to do a live poll and get a read on the audience and what everybody is thinking about. And so in her very nifty poll, really many of the same themes came up. So Christina asked the question, if there was one thing that could improve your child's health, what would it be? And intellectual disability is obviously one of the biggest word clouds, but I think some of that is really just was it because we type in different words? So I think seizures, seizure control, seizure free, no more seizures. I think those all align very much with what we saw originally with the Facebook poll. And so as I say, this is very helpful for us to think about going back to both our clinical research and our lab work as well. But towards the future, I think thinking in the context of natural history studies and understanding what is the clinical picture of individuals with CHD2 variants, there are a couple of themes that came out over the last day and a half. And I think one of them was really thinking about symptoms across the lifespan. So really, and again, it's an ascertainment bias. By and large, the largest number of individuals that we know about with CHD2 variants are more pediatric and teenage years. And I think over time that will change. I think it's hugely important, however, as David spoke about a little bit, and Danielle spoke about as well, is to start looking at what does the clinical picture of CHD2 look like in older adults. And so I think that's something that we need to be aware of. I think that we need to increase genetic testing in adults, push our adult colleagues to start testing in adults so that we can find these CHD2 patients. Also through research programs and perhaps also appealing to pharma and biotech to sponsor the genetic testing for adults, because at the end of the day, we really need to have good natural history studies across the lifespan to have good clinical trial evidence. And so along those lines, I think in the future, there is no one size fits all. There is no one study that I think everybody can enroll in and we will get the answers to our questions. And I recognize that that puts the burden back on you folks, and that in all likelihood, you'll be enrolling in multiple different clinical studies. And what I've really highlighted here are the ones that CCC has been involved in. So the RARE-X study, this is the study that Dr. Andrade spoke about yesterday. Citizen and Invitae is also enrolling, as well as you've seen over the last day and a half, and I think almost all of the families have visited with Dr. Mefford and her team in terms of enrolling in the bio. And so I think all of these things are very exciting, and I think that we need to maintain momentum and get more families to sign up. And so towards that, I'd like you all to, if you haven't already, enroll in the CHD2 census. So this is a link that is at the CCC website, where you can go and register your case with all of your information, and then CCC can get hold of you, share information like ongoing research studies, information about research, resources. It's a way for us to reach more people, which is really key in terms of understanding the number of people with this disorder and what does the clinical picture look like. And so you can see in this map, this is from the website, and you can see that there's a great scattering of people from all over the world. Hey, look, one in South Africa. I can do some work there. We have some CHD2 families in South Africa. So yeah, tell anybody that you meet with a CHD2 finding to register on the website. And then let's talk a little bit about lab science. And so you've heard over the last couple of days a lot about animal and cell models. I think this is super exciting that we actually now have these models. If you were talking to me 5 years ago, there was almost nobody working in this space. So I think this is hugely exciting. I think one of the real things that we need to focus on in the next 3 to 5 years is having measurable phenotypes. So right now, we really don't have that. We've heard 1 or 2 talks where there are maybe some changes in either the animal or cell models that we can start to measure, but we're really not there yet. And this, from the research perspective, really needs to be a big push in the next couple of years. And along with that, molecular biomarkers. Because once we get to a therapy, we want to be able to have something that we can measure. I think the second big goal, and where the lab science will be going, is we've heard how both too little, so haploinsufficiency of CHD2 is bad, but also too much CHD2 is also associated with neurological outcomes. And so what we're really going to have to focus on is really fine-tuning the amount of CHD2 protein. And that is going to be a challenge, but I think the exciting thing that we've discovered in the last couple of days is just the number of people that are working on different approaches. So I've listed some here in order to be able to fine tune this balance. So I think this is super exciting. We still have lots of work to do, but it's great that people are working on this. And so the goal then will be to measure, once we can fine tune this amount of expression, is see, can we rescue some of these phenotypes? And so another big goal that I personally have is I love to do collaborative science. And so this is something that I will push forward as well, to try and get researchers to work together. What can tend to happen is that we work in our own labs on our science, and that's great, but I think that we can achieve more faster if we're more open and collaborative. And so this is a real agenda item for us for the next, and the SAB in general, for the next couple of years. So how do we know what success looks like? And so thinking about the short and long-term goals. So in the beginning, Heather and I kind of outlined what are the long-term goals. And this is the roadmap. The long-term goal is to have treatments and cures, right? But I think what the other thing that's helpful is to think about what is the short-term success? What does short-term success look like? And so to me, I've been thinking about this in the context of the next 2 to 3 years. Where do we want to be that tells us that we're making progress towards this long-term goal? And so to me, I've broken these down into, again, the different categories in terms of identifying and defining the patients. I think having studies on many hundreds of patients right now, there's one study in the literature of just over 100 patients, and it's kind of aggregate data. It hasn't been done in a systematic way. So I would love to see in the next couple of years, at least 2 studies in large cohorts of patients. They're there to clear a picture across the lifespan, and at least 1 ongoing natural history study. In terms of animal models, like I said, I think our focus is if we have 5 robust, measurable phenotypes in 2 to 3 years, I will be overjoyed. Because this protein is tricky to work with. And so if we're there, I think that's a great mark of success. In terms of testing therapies, I think that, what's the word I want? Reasonably, what's another word for reasonable? Having complete blank. Realistically, that's the word I want. Realistically, I think in 2 to 3 years, if we're in preclinical models with at least 2 modalities that regulate CHD2 expression within a very narrow window, I will be incredibly stoked. And also a rescue of at least 1 measurable phenotype that's robust, as well as having candidate biomarkers. And then lastly, of course, we would love to involve more patients, enrolling in the CCC registry, enrolling in biobanking. But I think the other big role is really in terms of fundraising. None of this can happen without money, and that's just the reality of the world that we live in. And so encouraging you all within your communities to fundraise for CCC so that they can keep doing the amazing job that they're doing. And then lastly, of course, as well as fundraising, the other big thing is collaboration. And so I really think that this is key in terms of this has been a great example, right? bringing together the families, the researchers, the physicians. And so these are some cool photos from the last couple of days. And so I really think that this is the other thing that's key, is that we all just keep working together. All right, and so with that, I will end, and thank you everybody for your attention. And again,